part of the DNA, a region of DNA. A gene, again, is going to be what encodes a protein. So it's a gene that specifies a protein. And by doing so, it provides for a characteristic, right? So people have an eye color, right? The eye color characteristic results from the eye color gene. Although people have different eye colors, because there could be variations in the different eye color genes among different people. Okay, so the gene encodes the protein, thereby specifying a characteristic. All right? Question about what is a gene? We'll talk more about it to be sure. All right, so my question is, where does this take place? The gene encoding the protein. Where in the cell, how do you go from gene to protein? Okay, so don't answer. Here's the answer. Right, so where's the DNA? Of course, in the nucleus, right? So the nucleus stores DNA for the cell. Recall, right? The nucleus stores the cell's DNA. And so when it's copied into RNA, that happens where? In the nucleus, where the DNA is. Okay, so the first step, step one, happens in the nucleus. And here you can see how the RNA is a region of the DNA, right? Sort of. So let's say the DNA keeps going, that much is clear, right? It keeps going, keeps going, keeps going. It's just a part of it. I guess this is the end, right? It keeps going. But let's say this is all the RNA. Okay, this is the one RNA this is the gene here. It's copied, and here's the RNA. Okay, here's another RNA here that could be made from that gene. Here's another RNA that could be made from that gene, and so forth. Okay? Make sense? All right, so this RNA then exported from the nucleus to the cytosol. Exported to the cytosol, where it's then used to make protein with. So I guess that is decoding the RNA, making the protein, building the polypeptide. Making the protein means building the polypeptide in this case. Okay, who does that? Who decodes the RNA, makes the protein? Ribosomes. Ribosomes, if you want to make it plural, ribosome. Okay, good. All right, so how does the RNA go from the nucleus to the cytosol? To the pores. Okay, there we go. That's the connection. So the pores are important for providing a passage for the RNA to leave the nucleus and reach the cytosol. Okay? Yeah, that's what these pores are for, among other things. They're a way for RNA to leave. And understand again, the ordering of events that we spoke of, right? The DNA is here, and the copying part happens in the nucleus. The RNA is exported, and then the decoding part happens in the cytosol, right? So right there is an ordering of events that utilizes that compartmentalization. Okay? Also recognize the brilliance behind the strategy, right? So the nucleus is going to serve as a store of information, right? It's the administrative office where the information is and the decisions are made, right? What are we going to do? Well, then the instructions are sent to the factory floor, right? The cytosol, the factory floor, where then things can be executed, right? Where things can actually happen. Okay? Yeah. I mean, you're not going to take your library out of the nucleus to the factory floor, right? You make a copy. You make an RNA copy that then gets used practically to make the protein. There's a special name for the RNA actually, because it's shuttling the information right from the nucleus to the cytosol. What is it called? Messenger, right? The M is for messenger, right? The M there, messenger RNA. Oh, very good. Any questions? That's it. Enough. Question? Okay. Let's go on. Okay, and we'll talk about all this more down the road. Let's go on then to talk about endomembrane system, which again I said was the big dog, right? This is the big topic, I think, in this chapter. Let's embark on this now. And indeed, it does have to do with protein, this topic. Okay, so prologue, wow, yeah. Again, a question, where are proteins found in the cell? Where are proteins, the cell's proteins found? What's the answer? Okay, membrane I think is a great answer because we just said that. We said the cell membrane has proteins. We named a couple varieties, and then we went on to say that all of these internal membranes have proteins. Absolutely great answer. Are there any other answers? There's more. The cytosol. Yeah, the cytosol is full of different proteins. It's a seed, if you will, another seed of protein. Yeah. Anything else?
The ER.
The ER actually contains protein. Okay, let's just say it. Every compartment contains proteins. Okay, so I think the answer, if you're going to just say everywhere, it's fine, right? Where are the cell proteins? Everywhere would be fine, but if you want to be a little more specific, you could say every compartment, including the cytosol. Every compartment contains protein, and every membrane contains protein. That would be a little more descriptive. Okay? Where's the DNA to make all that protein in the cells? In the nucleus. There's only one answer to that. And in fairness, there is a second answer. I just contradicted myself. But we'll talk about it. So for the moment, the answer is nucleus. Nucleus. That's the DNA, by the way. Look at the DNA. That's all DNA. Okay? DNA is in the nucleus. Protein, 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 protein. But DNA to make the protein is here. Now that's a puzzle. If the DNA is there, then how does the protein, get to be here and here and here? Well, we started to talk about that a little. I think when we said the messenger RNA gets exported into the cytosol. You've already moved things now, right? You move things out into the cell, into the cytosol at least. But that's not a complete answer. So, what makes protein? Why don't we start with that? And then we can try to answer this as we go. What makes protein and how do they get distributed, right? So, as you both all told me, I think, ribosomes, right? Ribosomes. Ribosomes are the organelles with the job of making protein. And by that, it means they construct the polypeptide. They decode the mRNA, and then they construct the polypeptide. They join the amino acids together to construct the polypeptide. It's more descriptive when you say it that way. Construct the polypeptide. Okay, what is a ribosome? It's rhetorical. Let's take a look. What is that? Okay, I heard some different answers there. It's almost like, what is it not? I mean, oh my gosh. Okay. So, what is this? Anybody?
Protein.
Protein is an alpha helix. Yeah. What is this here? It's an alpha helix, which is a type of secondary structure, right? Of protein. And what is this? Everybody? It's another alpha helix, right? So we're seeing a lot of alpha helices, I think. Any beta sheet? Anybody? Maybe in here? Beta sheet? You guys have a better view of this than me, by the way. Okay, there's a lot of protein here. I mean, just looking at it, there's a lot of protein. What is that there? What is that? What is that here? Okay, DNA is a great answer, except it's wrong. No, I'm serious. It's a great answer because if you look at this and you don't think DNA, then I don't know what you think DNA is. It looks like a DNA here and here and maybe here and here, right? But it's not DNA. So what must it be? It's RNA. Yeah. The ribosome is made of RNA, and it's made of protein. So what is a ribosome? It is a structure with two parts, by the way, right? A large part and then a small part. It's a structure with two parts which constitute a macromolecular complex. This is not a macromolecule because that name is not big enough to describe it. It's so many macromolecules, right? It's so many macromolecules actually. So it's a macromolecular complex. And again, it has two parts we call subunits. They're just called the subunits of the ribosome. Each one, make no mistake, consists of several polypeptides. My 4-year-old likes to say so many if something is a lot. So many. It is so many polypeptides in the large subunit. So many polypeptides in the small. And there's RNA, but not to be mistaken with the RNA that gets decoded, the copy of the gene that gets decoded, which we call messenger RNA. So we need another name for this. We'll call it ribosome RNA. The RNA that constitutes the ribosome, makes up the ribosome, is ribosome RNA, or ribosomal RNA, or rRNA for short. Everybody has ribosomes, okay? Because everybody has what? Protein. Oh my gosh, yes. Everybody now. Every bacteria to euglena to frog to orchid to everybody has protein. So everybody has ribosomes, the ability to make protein. In eukaryotic cells, there are ribosomes in the cytosol. It's a pool of ribosomes, actually. There are ribosomes in the cytosol. But I think you already know there's also ribosomes attached to the surface of the what? BR. Yeah, there's ribosomes on the surface. So one way we can distinguish between these ribosomes, any particular time you see ribosomes in both places, the ribosomes in the cytosol are called free ribosomes. The ribosomes on the ER surface are called bound ribosomes. Bound. Okay, just to make a distinction between those two locations. And maybe a hint, maybe another hint as to how proteins get to be where they need to be in the cell. Right? Which is what this is. Okay? Yeah. What is that dichotomy about there? Could that be a hint about where proteins go and how they go there? Yes, it is. Okay? So now without further ado, that was all prologue. Let's talk about the endomembrane system. What the heck is that? So I think there's something rule in what's the science of language? right? There's a rule that says you don't define something with the same word, right? So in my PowerPoint, I think it was the endomembrane system is a system that the blah, 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 blah. That didn't sound, that sounded,
different.
I felt like I was violating the rule. So let's say collection, okay? So what is the endomembrane system? It's a collection of membranes, right? A system of membranes, a collection of membranes. Do I care you know the definition? No. I want you to understand what is the basis, though, for that being a collection of membranes. They're joined, but indirectly. Okay. What does that mean? It means to be joined indirectly. One membrane can break off a piece that then goes and becomes part of another membrane. One membrane breaks off a piece that then becomes part of another membrane. One membrane buds, the word buds, it buds off a piece that then fuses. That's the word, fuses with another membrane. Okay? And if you can do that, if you can bud off membrane, or if you can fuse with membrane, you are part of that system. You're part of the club, called the endomembrane system. Okay? And we just need a name for that piece then of membrane. It's called a transport vesicle. Transport vesicle. The piece of membrane budding and fusing is a transport vesicle. Why is it called that? Could it be used to transport something? Yeah. So that's the idea, right? So here they're showing pieces of the ER budding off and fusing with the Golgi, or Golgi apparatus. Pieces of the Golgi budding off and then fusing with the what? The cell membrane. Yeah. They're all part of the club. The ER, the Golgi, and the cell membrane are all part of this system because they have this capability of budding and or fusing with transport vesicles, right? And again, I wonder what these transport vesicles are about. They're transporting. What is that? What is this? It's not pepper. It's something that is being transported, right? So transport vesicle is a compartment. It is a compartment enclosed by a membrane. Okay? By a single membrane, lipid bilayer. It's a compartment enclosed by a lipid bilayer. That's why it's called transport vesicle, which again can move about between the components of the endomembrane system. Is it clear? Okay, it shouldn't be clear because this is a lot of information at this point. But if you're kind of there, then maybe it's okay. All right. I think the next question then is, what is the system of membrane? Can you be more specific? Who is part of this club? Again, the ER, we just said. The Golgi, we just said. Lysosomes are part of the endomembrane system. Food vacuole, very specific term, is also part of the endomembrane system. And the cell membrane. What does endo mean? Inside. Okay, the cell membrane is not inside, it encloses the cell. Nonetheless, part of the endomembrane system, because what? It's part of that system or collection of membrane that can bud and fuse transport vesicles. Okay? Question? Really? Okay, if not, let's get into it then. Beginning with the ER. The ER, as you know, is an acronym for endoplasmic reticulum, right? Endoplasmic reticulum. Again, a stunning micrograph of the ER is shown here. Now, if nothing else, it's extended, right? It's very expansive. It's spread out throughout the eukaryotic cell. Okay, it's everywhere. Okay, it's also made up of two parts, isn't it? This is all ER, left and right. Two parts. We've already mentioned rough ER, I think, right? This is the rough ER, and on the left is the what? Smooth ER, right? RER and SER respectively. RER and SER, for rough ER and smooth ER respectively. Okay, so let's just talk about each in turn. The rough ER is called rough. Why?
Ribosomes.
It's covered with ribosomes, right? That's all these little dots now. Again, are ribosomes, right? Yeah, and then if you can't make it out, you can always look at the cartoon, right? Okay, so rough ER, right? Up here, rough ER. And oh, way up here, smooth ER, right? Okay. Which one is connected to the nuclear envelope? Rough ER, right. So technically the nuclear envelope is also part of the endomembrane system because it's a physical connection. So you can be indirectly connected in the manner we described, but you can also be physically connected. And so the rough ER, smooth ER are physically connected to one another. The rough ER is connected to the nuclear envelope. So technically it's a part of the endomembrane system. Okay, again, rough ER so named because of the presence of ribosomes. All these are ribosomes, right? And you can see in the cartoon here. So I don't even need to do anything. What does the rough ER do? What does it make?
Proteins.
Proteins, absolutely. It's covered with ribosomes. Of course it does. Right? The question is, what happens to that protein? That's worth considering. Now, so what happens to the protein? Again, decades of cell biology, I can just tell you in 5 seconds. The protein enters the ER. The protein we know, made by the bound ribosome, enters the ER. It is threaded across the ER membrane. Threaded is a good word, I think. Threaded into the ER. Okay? I always feel the text doesn't show this very well. Your text is great, but it doesn't show this. Threading. So I got this figure from a graduate textbook. I am confident you will make short work of it, no problem. All right, so let's look at it together. Let's look at this together. This figure is showing the ER. Okay, this is the ER. And this is the ER membrane. Again, a single membrane enclosing the ER. Now you can see there's a space within, an aqueous space. So, what's that? Parked on the ER surface. It's a ribosome. There you go. And what's it got there in its jaws?
The
mRNA. Yeah, shown in blue here. That's mRNA. It's also labeled. There you go. Okay, so the question again is what happens to the protein that's made by these? And I just feel like I can't just tell you, you're going to see, right? So here it is. Here's the protein now, the polypeptide, and consider this a time lapse. Okay, so time 1, time 2, whatever that is, and now we're decoding the RNA, right? Decoding, and oh, look at the polypeptide being made, right? And then further along, we decode, and now look at the polypeptide. Okay, the ribosome is done. Okay, so where's the protein? Inside the ER. Again, threaded across the membrane into the ER space, or it's called lumen, into the ER lumen, the interior of the ER. Okay. All right. A question about what's happening here. All right. So again, years of research has shown this, but we know this to be the case. All right. So one more thing about the ER. Somebody said earlier when I was asking where the proteins of the cell are, somebody said the ER has proteins in it. That's true. There are proteins in the ER. Well, I mean, I guess this is there now. This protein entered. Moreover, there's a family of proteins that live in the ER called chaperones. Chaperone proteins live inside the ER. Chaperones. They're a type of protein. All right, what are chaperones? This seems kind of random. So there's a concern, right? When you make so much protein all in the same place. And just look, everybody, for a moment. Just look at, this is a real photo now. How much protein synthesis must be happening here? This is nothing but ribosomes. So this is barely doing it justice. So I said this is a time lapse, but and now this could also just be free ribosomes, right? They are, I mean, the ER surfaces wall-to-wall ribosomes, which means you've got all this protein entering at the same time. What's the problem with this strategy of making protein?
Build up.
So, well, how would you describe the chemistry of a protein? What is a protein's chemistry like?
Charge.
I mean, what
Polar.
is it not like, right? We said the amino acids could be polar, yeah? They could be charged, or they could be what? Non-polar. Okay, so these are the three families of amino acids. In other words, proteins are both hydrophilic and hydrophobic. So it's like a phospholipid. It's both hydrophilic and hydrophobic. These are incredibly sticky molecules, right? They're hydrophilic, so they have charge, they have partial charge. They're also hydrophobic, so they stick together like fat. They do everything. And you're telling me, and the protein has to go like that, right? A protein, I mean, that chemistry is important because the protein goes like that, right? Its chemistry makes it do that. But if you're right next to another protein as it's being made, what could happen here? They could stick together. Yeah. So we need something that will get in here and prevent undesired interactions between the proteins, prevent them from interacting. That's what a chaperone is. What is a chaperone, by the way, at a dance? Somebody that
Watches. Separate.
You're so shy, right? Prevents undesired interactions from happening. Folks, this is why they're called chaperones. I'm not kidding. Yeah, prevent everybody from hooking up, right? That's what it is. Chaperone. Biologists know things too, guys. Okay, so, all right. So, chaperones are a type of ER proteins that allow the proteins that are made at the ER to fold correctly. They allow them to fold correctly by preventing them from interacting with other proteins before they're folded. Once they're folded, it's fine. But before then, there's a problem. Okay? Question about chaperones? Okay, I have a question. What happens to the protein, right? Oh, question, please. Just ask the question. I'm not going to see you. There's a thing for that.
What sets apart a chaperone protein from a regular protein?
So a chaperone is a good question, right? What is it that allows a chaperone protein to do this job and then not get tangled up itself, right? So a chaperone protein, I mean, as I understand them, they're tube-shaped proteins, right? So they're literally kind of hug the protein as it's being made, allowing it to fold, and so it can't interact with anybody else. Okay, so I think it's fair to think of it just literally as something that kind of gets a hold on polypeptide as it's folding. Okay, yeah, great question, nice. Okay, my question was maybe as interesting. How, or rather, what happens to protein after it's made, right? What is the deal with making all this protein? So please understand there's multiple possibilities here. So a protein that's made on the ER surface could stay in the ER and just be an ER protein, right? We've now explained where chaperones come from. They're ER proteins that you know how they're made, right? They're just inserted into the ER and then they're done. But proteins made at the ER surface could also leave. They could also leave the ER. And at that point they would go where? Well, it's a little bit limited. You can only go to other parts of the endomembrane system. That's the limit, right? You can only stay within the endomembrane system. And you travel by what? Transport? Vesicle. Yeah. Let's say it. Transport vesicles, transport proteins. There we go. There we go. There's proteins in this vesicle, right? And then some of them got stuck in the membrane. I wonder what's going to happen with these proteins. Some of them are membrane sort of affiliated proteins versus these that are inside and soluble within the transport vesicles, transport proteins. All right, smooth ER. In our remaining time, let's talk about smooth ER. No ribosome. So what does it do? I'm just going to tell you again, years of cytology, biochemistry. They make lipids. They make lipids, and they really make all of the types that we talked about. They make the variety of lipids. So the smooth ER is all about making lipid, actually. All right? Another function of smooth ER is it has enzymes that can detoxify harmful substances. So detoxification of drugs, essentially, is a function of the smooth ER. Another function is storing glycogen. So these last two are actually more kind of cell type specific or more tissue specific types of functions, and not all organelle functions are performed by every organelle, if that makes sense. So, for example, these last two functions are going to happen where in your body?
Liver.
In the liver, right? So liver cells would be expected to be rich in smooth ER. Liver cells must be chock full of smooth ER. So what type of cells have a lot of smooth ER? Would be liver cells, right? That makes sense. Make sense? Yeah, I think so. So, in conclusion, the ER makes both lipid and protein, actually, right? Now, what cell structure that you know of consists of both lipid and protein? The membrane. The cell membrane, actually, is made of both lipid and protein. And remember, another feature of life is growth, right? All cells grow. So when a cell is born, the process of cell division, right? It grows. It grows. It grows. And the way it grows is because it is fed vesicles that are made by the ER. Okay? So one job of the ER is to make membrane, literally to make membrane which is made of lipid and protein. The ER makes lipid and protein, and so it makes the cell membrane. Alright, so ER products are either going to remain in the ER or they will leave. Where do the ER vesicles go? We said it earlier in the lecture. They go to the Golgi, right? And this is like the most famous route. It's not to say there could be others, but the most famous and well-characterized route of vesicular trafficking is from the ER to the Golgi. And I think we should take it up there on Wednesday. Questions? Nice work, folks. Have a great week. Yeah.